Remember that from a DNA biochemistry lab technique perspective, we can analyze and isolate nucleic acids for study, which has some clinical applications. Specifically, remember that we can separate molecules by size and shape via gel electrophoresis. For RNA and DNA, we just need regular old native page, since the backbone is so negatively charged that the nucleic acids always move towards the anode, which is positively charged. Once we have separated our molecules, we can identify what we're isolating with blotting. We've used Western blotting in other lectures to identify proteins, but specifically we can use Southern blotting for DNA and Northern blotting for RNA. What specifically is happening is that we're using the gel electrophoresis to separate our molecules and then transferring our sample to another surface, which is nitrocellulose, and then immobilizing our samples with UV light. We then introduce a labeling strand or a probe sequence of DNA or RNA to the separate strands, and our specifically designed probe will then bind preferentially to what we're looking for. So for RNA in northern blotting or DNA in southern blotting, we have to build this complementary RNA or DNA strand that is bound to a reporter molecule like a colored light or radioactive signal, so we can visually see it when it is bound. The key then is to design your probes to be able to create a complementary strand based on base pairing to the target sequence. So just making sure that you're keeping in mind the directionality of the 5' to 3' or 3' to 5' strands. There's one final difference between southern and northern blotting in that usually our DNA sample is double-stranded, so specifically in southern blotting. So first you usually need to add this extra step of denaturing the DNA to get it to be a single strand to be able to even bind the probe and reporter. Another important biochemistry technique we use all the time is centrifugation. Not only used for nucleic acids, it involves taking a fluid sample, like the blood, and then separates its components by spinning the tube at very high speeds. What happens is that the substances with higher mass, that are usually denser and with less surface area, appear on the outside of the tube, while smaller, less dense substances appear closer to the center of the centrifuge. After allowing to settle, what you end up with is two layers: the supernatant, which is this liquid, less dense phase, and the pellet, which is a solid, dense substances contained within, allowing separation and then isolation via chromatography. We of course discussed chromatography in detail in organic chemistry, but I will link to that video clip below. But let's now discuss another type of DNA-based biochemistry technology in DNA sequencing and the polymerase chain reaction, or PCR. DNA sequencing, of course, refers to a technology that is designed to determine the DNA sequence by base pairing. Specifically, something called the Sanger method was developed in 1977 by Frederick Sanger, an English biochemist who received the Nobel Prize twice. In this method, we use the DNA we are trying to sequence, the template strand, denature it to obtain a single-stranded DNA sample, and add a primer to allow DNA polymerase binding and base pairing of the template strand by manually adding nucleotides into the system. Instead, however, we also use these nucleotides called ddNTPs, which are labeled with no 3' hydroxyl group bound to a fluorescent reporter, giving a unique color for each base pair. So for example, ddCTP may be teal colored and ddATP may be red. What specifically happens is that the DNA polymerase is adding dNTP, so regular nucleotides, until it randomly chooses a circulating ddNTP in solution to incorporate. And once it does this, the DNA polymerase falls off due to chain termination. There's no 3' hydroxyl group and DNA polymerase falls off. The unfinished strand then is labeled with a color, but now we only know one base pair and can determine its position through gel electrophoresis for length determination. To find the full sequence, you have to run this reaction with hundreds of DNA sequences in question and multiple ddNTPs of a single variety. So imagine that you'll end up with many strands of various lengths depending where the ddNTP was incorporated, and then you can find all of their lengths. You repeat this experiment using all 4 ddNTPs, so A, T, C, and G, and then have every single base pair mapped. Running them side by side on a gel, you can then determine their position. See in this graph above, the distance traveled will be inversely proportional to their length. Remember, the longer these strands are, the less they move easily through the gel matrix. So see in the picture above in the third section, you have the gel matrix run with all of the strands. The T nucleotide at the bottom is the farthest traveled, so it must be the smallest length or base pair 1 at the 5' end of that DNA sequence. And then you read the sequence up the chain, obtaining the final sequence. Remember, this sequence we've produced is the template strand sequence. To find the unknown sequence we want, we then need to simply predict the complementary sequence. So to our bottom right here, if we isolate TAGGCTCA, we will predict our unknown sequence to be TGAGCCTA if you're reading it 5' to 3'. But importantly, how did we obtain hundreds of copies of our DNA sequence in question to even run the Sanger sequencing? We probably did it through a very clinically important technique called the polymerase chain reaction, or PCR. PCR simply allows us to use DNA polymerase to multiply our DNA strand by thousands of times to create more copies. You don't need ddNTPs, of course, just primers, which need to be designed, heat-resistant DNA polymerase, dNTP, so regular nucleotides, and our double-stranded DNA. The heat that we're going to add allows DNA denaturing so that the primers can bind, and then the reaction can begin, hence why we need these heat-resistant DNA polymerases. Primers bind via annealing once the temperature is lowered again, and then temperature is risen again, allowing the DNA polymerase to elongate the chain, extension or elongation. The binding of complementary strands after elongation is called hybridization, and at the end, we have double the DNA we started with. Now 2 double-stranded DNA strands. We can then rinse and repeat, obtaining 2N DNA for each run, N of PCR. Quantitative PCR, or qPCR, gives us a more accurate idea of how much DNA we're producing using a fluorescent probe that binds double-stranded DNA. The amount of fluorescence then correlates to the number of completed double-stranded DNA strands. Alternatively, reverse transcriptase quantitative PCR, or RT-qPCR, of course involves turning RNA into DNA, where reverse transcriptase first turns the RNA into complementary DNA, or cDNA. We then multiply the DNA by PCR and probe the dsDNA to quantify. Importantly, cDNA, or complementary DNA, from RT-qPCR may be more interesting to study, since it only contains exons, being a reverse product of mRNA, which has already been spliced. By performing RT-PCR, we can therefore build something super cool, which is a cDNA library. cDNA from this library can be inserted into a host cell and then used to produce a specific protein on a large scale. Some other terms that are useful to know are recombinant DNA, which refers to DNA whose nucleotides have come from 2 different sources. Hybridization, as we've said, refers to the joining of 2 DNA strands because of complementary base pair matching, and this could be used, for example, in a paternity test. DNA libraries are libraries of DNA sequences, whereas expression libraries refer to our cDNA libraries, which are coding only, more useful for production and expression of proteins. You can perform hybridization on a living organism in so-called in situ hybridization. When fluorescently labeled, we call it FISH, or fluorescent in situ hybridization, by designing a complementary DNA probe. Importantly, Sanger sequencing in RT-PCR is often used to create circular DNA, or plasmid DNA, used in bacteria, which is replicated independently of chromosomal DNA, another DNA reservoir, with key features of plasmid DNA being multiple cloning sites, or MCS, that are short DNA fragments that can be cut or different at specifically different sites with restriction endonucleases, as we'll discuss. Inserting foreign DNA into a plasmid is an example of recombinant DNA. Plasmids can be inserted, replicated, and transcribed in bacteria, so-called a transgene, or exogenous gene, and the proteins expressed thereafter we call recombinant proteins. Now, ELISA is a very simple DNA technology that is used in modern medicine to detect the presence of specific proteins or molecules in the body. I did a research project on heparin-induced thrombocytopenia in my training. We use an ELISA hit assay to detect the presence of a heparin PF4 protein complex that indicates the presence of active disease, where a patient's body is creating very thrombogenic heparin aggregates in the blood, probably as a hypersensitivity reaction to heparin. What the ELISA actually does is fix the antigen on a surface surface, if it exists at all in a sample like serum, and washes the fixed surface with an antibody which is known to be able to bind to the antigen of interest. If it is bound, we can tell binding has occurred in direct ELISA if we then see a fluorescent reporter go off, since a reporter is bound directly to that primary antibody, or after one more step in indirect ELISA, where a second antibody is washed over the first, which can bind to the primary, lighting up if this binding occurs. We also use ELISA, for example, for our detection of syphilis. ELISA can also work quantitatively if you use sandwich ELISA, where two antibodies are bound, but both this time directly bind the antigen, instead of the second antibody simply binding the first antibody. And the strength of light or reporter signal in sandwich ELISA can be correlated to the quantity of the antigen bound. From a molecular biology perspective, we can harness bacteria for the replication of DNA, since bacteria can carry extra chromosomal exogenous DNA in the form of plasmids, as we've discussed. Bacterial transformation involves the uptake of environmental DNA, incorporating it naturally into its genome. This allows bacteria to potentially survive threats that other bacteria nearby have died from. But we can also enact transformation in the lab setting by giving bacteria a plasmid of circular DNA with a gene of choice, allowing the bacteria to transcribe and express DNA of our choosing. Another important technique relevant to this is molecular cloning, which refers to the replication of a specific DNA fragment by multiplying it inside a bacterial vector via a plasmid. Molecular cloning then really replaces PCR in this way. In order to insert this DNA in a plasmid, we need to use restriction enzymes or restriction endonucleases, which can cut DNA at palindromic sequences around 4 to 8 base pairs long, reading the same forwards and backwards, creating short single-stranded regions. Specifically, the palindromic sequence is double-stranded, so one strand will read forward and the complementary strand will read the same going backwards, 3' to 5'. Our restriction enzymes recognize these unique sequences and create a small DNA single-strand break, creating two sticky single-stranded DNA regions, one on each side, that can rebind then very easily. But what we do specifically is to make a plasmid is to place the palindromic sequence at the ends of our complementary DNA regions. Think cDNA is the DNA we're inserting, and so you put palindromic sequences on both of its ends, and then you use endonucleases to make those ends very sticky by creating single-stranded breaks, and then allowing the sticky ends to seal, creating circular DNA that seals inside a complementary DNA sequence that can be added then into a bacterial colony through transformation. One way of confirming transformation is by adding a second gene that has a notable function, like antibiotic resistance. If the cDNA sequence was taken by the bacteria successfully, it will also then have taken in an antibiotic resistance second gene that we snuck in there, and resistance then confirms that we've integrated our DNA. One other way we can confirm plasmid presence of our gene is through including a reporter gene like the LAC-Z gene inside the same plasmid. When translated, the reporter gene will produce a detectable signal. So this is a way of detecting successful transformation once translation has been through. The LAC-Z protein is an example of such a reporter gene, and notice in the diagram to the left, one way of checking for confirmation our cDNA has been incorporated is by adding the palindromic sequence in the middle of the reporter gene. The cDNA then will be incorporated into the middle of the LAC-Z once the sticky ends anneal with the separate antibiotic resistance gene in the plasmid. Once bacteria have been given this plasmid and exposed to antibiotic, bacteria which are showing color after being exposed to the LAC-Z substrate do not have our cDNA, since our cDNA should be interrupting that gene being placed in the middle of it. If we add the LAC-Z substrate and no color change occurs, we have incorporated our cDNA and can grow this colony. So you can see there's a couple ways of confirming, either through the presence of a reporter or the absence of a reporter, can both help you find out if we've successfully transformed some DNA. DNA ligase specifically is the enzyme that can anneal kill the new DNA fragment inserted into the plasmid. Although plasmids with recombinant DNA can be produced via generation of cDNA and cloning, a major problem is which host organism to use. Bacteria are the first choice due to easier culture conditions and faster growth, although they don't have a spliceosome. cDNA fixes this problem by generating DNA only from post-spliceosome messenger RNA. Since bacteria also lack post-translational modification like glycosylation, for example, yeast are used for certain essential eukaryotic proteins. In other cases, insects or mammalian cells may be necessary. And lastly, to improve expression, promoters may be artificially used inside the plasmid upstream of the gene to increase activity and expression. So now let's take another practice passage to confirm what we've learned so far about DNA technology. Researchers have shown that radiation therapy has significant immune modulatory effects and is capable of unleashing potent anti-tumor CD8 positive T cell responses. Radiation therapy has been shown to enhance the efficacy of various T cell targeted immunotherapies that improve antigen specific T cell expansion, T regulatory cell depletion, or effector T cell function. The ability of radiotherapy to enhance adaptive immune responses has further been highlighted by the PD-1 PD-L1 checkpoint blockade in preclinical models of CD8 positive T cells. Several proteins in this checkpoint blockade are activated by RVF3, a small binding protein indicative of radiation-induced damage, including the TMR alpha inhibitor, which is activated at nanomolar concentrations of RVF3. While interest in abscopal effects, or those observed outside of the field of irradiation, have increased in part due to the observation that radiation can act as an in situ vaccine, researchers have recently determined that combinational radiation and checkpoint blockade therapy requires pre-existing T cell responses to control tumors. Given the strong interest in using existing therapies such as radiation to enhance alpha PD-1 and alpha PD-L1 responses in human cancers, it is critical to understand the mechanisms by which RT is improving outcomes to better inform the treatment of patients. In this study, researchers set out to determine in checkpoint inhibitor resistant models which components of radiation are primarily responsible for overcoming this resistance. In order to model the vaccination effect of radiation, researchers used a Listeria monocytogenes based vaccine to generate a large population of tumor antigen specific T cells, but found that the presence of cells with cytotoxic capacity was unable to replicate the efficacy of radiation combined with the checkpoint blockade. However, researchers did demonstrate that a major role of radiation was to increase the susceptibility of surviving cancer cells to CD8 positive T cell mediated control through enhanced MHC1 expression. Researchers also discovered that treatment with interferon gamma, IFN gamma, increased MHC1 expression as shown in figure 1 and improved susceptibility of cancer cells to control by antigen specific CD8 positive T cells. Researchers also observed a novel mechanism of genetic induction of MHC1 in cancer cells through upregulation of the MHC1 and its transactivator SI and found significant differences in the induction responses of two cell lines after treatments with radiation and IFN gamma. In order to perform this assay, researchers first constructed a fusion of GFP and the model antigen SIY to determine the level of expression of SIY. Researchers then examined a potential relationship between this response and the alpha PD-1, alpha PD-L1 response. The data supports the critical role of local modulation of tumors by radiation to improve tumor control with combination immunotherapy. So a very verbose passage and a lot of fluff contained here. And here are our figures. Don't spend too long looking at them. Of course, when you see a problem like this, a lot of acronyms, you essentially see this big response to interferon gamma with the induction of H2K with all three cell lines, so there's nothing more to look at with this figure on the top. And then on the figure on the bottom, with more radiation, you get this bigger response, which is expected, and then the interferon gamma is on the last figure there. And now let's try some practice questions. As usual, a long verbose passage and then a lot of questions that don't really need you to understand the details of everything in the figure or even in the text. So let's just start with question 1. The researchers discovered that they accidentally mixed up two cancer lines, OT1 and 2C, but they were able to identify 2C because its genome contains a unique, highly repeated DNA sequence, and then it's given here. What sequence could the researchers use in their southern blot probe to identify this cell line? So try for yourself first. Remember, southern blot helps us identify DNA, and you could use a complementary strand to bind to it once it's on the agarose, and then label it with a reporter to confirm that you've actually got the DNA sequence. And so because we're given our DNA sequence in 5' to 3', we're looking for a sequence that's complementary to this sequence in DNA, so it doesn't need to have uracil, in the 3' to 5' fashion. So we're given all four answers, 5' to 3', you're looking at them backwards, and the fastest way is to just look at the end here on our 3' end of our sequence that we're given, and it's CCC, so we're looking for GGG, and the only answer that gives us that is C. Question 2, what was the role of GFP in this study? Try for yourself first. In the text specifically, we were specifically told in order to perform this assay, researchers first constructed a fusion of GFP and the model antigen SIY to determine the level of expression of SIY. So SIY was the antigen of interest, and GFP, if it was able to do anything that was fused to SIY that let us know how much we expressed it, it must have been the reporter enzyme to indicate the level of translation of SIY in our case. Question 3, researchers have discovered a method to synthesize the NLRC5 cell receptor, which conveniently has an extremely short amino acid sequence. The researchers vaccinated mice with the receptor and isolated an antibody that is highly expressed in these mice compared with controls. Which technique should researchers use to determine if the antibody expressed effectively binds to this receptor? Try for yourself first. So specifically, we're asked to determine a technique that would help us decide if the antibody already expressed by these mice effectively binds to its receptor. So we need something else that can help detect when an antibody is actually bound to a receptor. So you know we're talking about ELISA. In direct ELISA, the antibody expressed by the mouse would have to be labeled for us to be able to detect it. So in this case, we use indirect ELISA, using a secondary antibody that attaches to a primary antibody that's bound to some antigen. And when all three happen, we get this reporter. PCR only helps us multiply DNA, southern blot helps us recognize DNA, and sandwich ELISA is a quantitative method of ELISA. Question 4, researchers have isolated a particularly robust strain of OT1 cancer cells that exhibit a high level of potency immediately after radiation therapy. Researchers want to look for the mRNAs of 3 different cell cycle activators expressed in this line with several other OT1 lines. What technique should researchers use? Try for yourself. So again, we're looking for mRNA. So we need a DNA technology that helps us identify RNA. We already know that northern blot is our technique that we generally use to identify. We don't use it to separate RNA. We use gel electrophoresis or centrifuge. But once we have it isolated, then we actually identify it with northern blot. So in this particular case, A wouldn't make sense. B, reverse transcriptase followed by PCR and gel electrophoresis wouldn't help us identify RNA. C definitely would help us grab these mRNAs, and reverse transcriptase followed by PCR certainly also wouldn't make any sense. Some more questions. Question 1, which of the following DNA sequences is most likely to be recognized by a restriction endonuclease? Try for yourself first. So remember, what do restriction endonucleases cut and what are they able to recognize? They are able to recognize palindromic sequences. And so that means that it will read, because these sequences are always double-stranded, you're going to have the strand read forward 5' to 3', the same as its complementary strand will be reading 3' to 5'. So in this particular case, you're trying to read it and decide if the complementary strand would read the exact same going backwards. So starting with answer A, we know that it would start with TT, and so it's already not palindromic. We can tell that its complementary sequence wouldn't read the same. Looking at answer B, we have ACA. We would expect that the complementary sequence would be ACA and then TGT, and so that we actually expect that would be the palindromic sequence. Question 2, which of the following techniques would you most likely use to isolate a protein with a net positive charge plus 35? Is it a size exclusion, gel filtration, anion exchange, or cation exchange? Very simple. This is a cation. To isolate them, we can use cation exchange. I've accidentally already given you the answer, but it's very simple. We don't use ddNTPs in PCR. What are we using ddNTPs for specifically? What DNA technology? That's Sanger sequencing, a DNA sequencing technique. We only need dNTPs for PCR, but we also need heat-resistant DNA polymerase because we heat them to separate the double strands before we reanneal them. Double-stranded DNA is the normal substrate for PCR, and then 2 primers are generally needed. And question 4, a bond formed between which of the following amino acids is disrupted in reducing SDS page? Hopefully you recognize the only reason to use reducing SDS page is to disrupt disulfide bonds, which are produced by the cysteine residues of proteins. We should also mention some other DNA technologies that help us analyze gene expression. Thus, of course, this means the presence of proteins present in a cell or outside of a cell which reflect successful transcription and translation. Really, these are protein detection techniques, and we have a few. Say that you want to see where a protein is being expressed inside of a cell, i.e. the destination of a protein. This could be done, as we've previously hinted, through FISH or fluorescent in situ hybridization, tagging a transcript with a probe, and then the complex creates a signal when it is expressed, and this can be visualized. Immunohistochemistry involves looking for specific disease-related proteins, such as HER2 receptors, sort of like an ELISA. You add an antibody designed against the target protein and a labeled second antibody. If the first antibody binds, the second will, and then you'll see a signal, inferring the presence of the antigen in question. Flow cytometry is a diagnostic lab technique done daily in the inpatient setting, which stains single cells looking for protein markers by washing with specific fluorotagged antibodies. Labeled cells then will emit light when passed through a beam of light. For example, we detect CD4 positive T cell counts with flow cytometry in order to predict the extent of immunosuppression in uncontrolled HIV. But how can we tell the function of a gene if we don't already know the protein it expresses? One way is through the evolutionary comparison, taking common sequences with known function in bacteria and in yeast, and then inferring function in humans, but also through protein domains, where we compare the protein expressed to other similar proteins. For example, the presence of a protein domain called zinc fingers, which are common in transcription factors, or plextrin homology domains, important for lipid binding and signaling pathways. Gene network mapping allows us to find which proteins bind to an expressed protein via protein interactions, which help us elucidate a cellular signaling pathway. And we can also infer function by cellular localization in FISH. Another important technique is immunoprecipitation, where protein binding partners are precipitated out, helping us find out which proteins will bind together and thus must be related in a pathway. Proteins are separated after adding microscopic beads, first through centrifugation and then Western blot, as we know, or mass spectrometry. Now, stem cells are a very interesting topic simply because they open an important potential avenue for amazing treatment modalities through the administration of autologous, which is self, or allogenic, which is donated cells that can differentiate and specialize into cell types which can either replenish depleted cells in the body, for example, in the meniscus, white blood cells, or others, or provide direct immediate activity in the body against a disease. So, for example, with CAR T cell therapy for CMV disease or active malignancy. What all clinicians and healthcare providers must understand about stem cells first is that stem cells are not all created equal, and that 2 different so-called stem cells can actually be very different in their ability to differentiate in the cell you might want. As such, if someone said they received stem cell therapy, they may have received a very specific and non-differentiable cell that may or may not have been helpful, in contrast to the widely differentiable pluripotent stem cells that we could harvest, for example, from an embryo inner cell mass, with great ethical debate on the morality of that process. Further, even embryonic stem cells are not completely differentiable. The only stem cell that is truly a blank slate and can become any human cell are zygotes or blastocysts, only at the moment creatable through fertilization of an ovum via the action of a sperm. These we would call totipotent stem cells. The next most broad we call pluripotent stem cells. An example are embryonic stem cells, only harvestable currently through the direct extraction of the inner cell mass cells, which really is taking cells from a zygote, which unfortunately destroys the inner cell mass in the process, killing the growing zygote. Embryonic stem cells can become any human cell except for extra embryonic cells like a placenta or the umbilical cord. The stem cells you have heard of are probably much more narrow in scope, often what we call multipotent stem cells, which can differentiate into a variety of cells within a specific lineage, but not any human tissue. For example, hematopoietic stem cells can become any white blood cell or platelet or red blood cell, and mesenchymal stem cells can become skeletal tissues, cartilage, and bone marrow, for example. These are harvestable through umbilical cord blood, which appears to be less of an ethical issue. Although stem cells are exciting for multiple reasons, the regrowth of tissue from pluri or totipotent stem cells is not a Health Canada or FDA approved practice due to ethical reasons at this time, but something to look forward to in the future. Related to stem cell activity is the lineage and cell specialization behavior of stem cells. For example, the maturation of a hematopoietic stem cell, a multipotent stem cell, into an osteoclast, for example. Cell specialization occurs in two discrete steps. First, determination, where a cell irreversibly commits to a cell line, and then differentiation, where it undergoes the initial biochemical and structural changes to carry out functions in that new cell type. But how biologically does a cell get the signal to become a new cell type? One way is through a gradient. During mitosis, an uneven distribution of mRNA during the process of mitosis may drive differentiation due to selective transcription, but more commonly it is because of cell signaling molecules that trigger differentiation through substances we call morphogens, which trigger the expression of pathway-specific genes as transcription factors. Any cell that releases morphogens on other cells is called an inducer. The receiving cell is the responder. Specialized cells typically need to be housed in a specific location suited to their needs. During development, the migration of cells to specific anatomical locations, for example, thyroid cells to the thyroid, occurs during developmental cell migration, with tissue-level cells forming a community of similar cells to form a body tissue. Neurulation is another example of this, with movement of neural crest cells, and errors during this process can cause various developmental disorders and result in intellectual disabilities, among other problems.